So hello everyone. So today I'm gonna discuss to you my REN R580 course project, and I am John Maverick De Leon, and my project is about assessing soil carbon stability in Alberta using mid-drift spectroscopy. Okay, so one of the vital functions of soil is that it can act as a carbon pool or sink that can help mitigate the rapidly increasing climate change and global warming. So carbon sequestration and carbon stability are two key concepts in this context. So soil carbon sequestration is the process of removing CO2 from the atmosphere, primarily mediated by plants through the process of photosynthesis and storing it into the soil. And then on the other hand, carbon stability is the stability or the resistance of the organic matter to decomposition in the soil, even with the presence of various factors or disturbances that can negatively affect carbon sequestration. So these factors that can affect carbon stability includes the widely known farm management technique, which is tillage. Then different slope and different depths can also affect carbon stability. So considering these factors, it is crucial to assess carbon stability to better understand how they influence carbon dynamics in soil. Okay, so for my research objectives, first I want to determine if tillage, slope, and depth have effects on the carbon stability of archived soil samples based on the absorbance spectra produced from mid-drift spectroscopy. And lastly, I want to know the carbon stability of most of the archived soil samples from the SQMP dataset that is collected last 2019 to 2020. So in this study, my null hypothesis is carbon stability as indicated by absorbance is not significantly affected by tillage, slopes, or depths, and my alternative hypothesis is carbon stability as indicated by absorbance is significantly affected by tillage, slopes, or depths. Okay, so for the study site, Soil Quality Monitoring Program or SQMP have archived soil samples from 2019 to 2020 that was collected in the different ecoregions and eco-districts in Alberta, Canada. Then 174 soil samples were collected from each eco-districts from each different slopes, low, mid, and upper slope, and depths 0 to 15 cm and 15 to 30 cm. And then in the map, the different colors represent the different ecoregions. So this is one ecoregion, this is one ecoregion. And then the number within the ecoregions represents the eco-districts. So for this ecoregion, this is the eco-district, and this is the eco-district. And then in the SQMP dataset, most of the samples were collected in this part in each eco-districts, and also in this part in each eco-districts and ecoregion. Okay, so in this study, mid-diffuse reflectance infrared Fourier transform or mid-drift spectroscopy was used to run the soil samples with a spectral range of 4,000 to 400 cm-1. Bulk soil samples were used in this study to assess the carbon stability. Then wet sieving with abrasion was implemented to the soil samples using 53 sieve. Then each of the soil samples was segregated into triplicates from each eco-districts per slope and depths before inputting it to the machine for analysis. The soil samples were scanned by using the Resolutions Pro software with 64 scans and resolution of 4 cm-1. After running, 1,869 wavelengths from the soil samples were produced. Okay, so now let's go to the results. So from 1,869 wavelengths, we will be just focusing on these wavelengths right here, these 28 wavelengths right here, because based on the previous literature or existing literature or studies, these wavelengths are the most important wavelengths to use to determine or to assess the carbon stability in soil. So in each wavelength, it corresponds to different functional groups that have different carbon stability rank. So in this graph about carbon stability distribution of the archived soil samples, you can notice that most of the absorbance from the soil samples was detected in the wavelengths that have functional groups that have moderate carbon stability. So it was represented by the violet or purple bars right here and also right here. So in this graph, we can assume or we can say that most of the absorbance of the soil samples or most of the soil samples have moderate carbon stability based on this graph because there are 13 wavelengths that belongs to the moderate carbon stability group compared to the high carbon stability group and low carbon stability group that have 9 and 6 wavelengths respectively. Okay, so since most of the absorbance from the soil samples was detected in the wavelengths with moderate carbon stability, we will focus on that stability group. So in this slide, it shows you the box plot for moderate carbon stability, and all of the absorbance mean right here belongs to the wavelengths that have moderate carbon stability. So as you can see, different depths, slopes, and tillage treatments have no statistically significant effects with p-value greater than 0.05 to the absorbance spectra of all the wavelengths with moderate carbon stability. The medians and the interquartile range boxes remains relatively stable and consistent with slight variation in each treatment groups. So the medians are the lines inside the interquartile range. So as you can see, the medians and the interquartile range or IQR boxes remain stable or consistent in this part of the graph and in the range of 1.2 absorbance means to 1.4 absorbance mean. So it remains stable in this range of absorbance mean. And as you can see, in each treatment groups in depth, slope, and tillage ranking or tillage level, it also remains stable in that range, like this one, and then in this one. So this indicates no significant effects to the mean absorbance spectra of the archived soil samples from 2019. Then after doing the three-factor ANOVA, it gives us this table, this ANOVA table. So as you can see, the p-value of the predictor variables, the depth, slope, and tillage, is greater than 0.05. So that means that depth, slope, and tillage rank do not have significant effects to the absorbance of the soil samples or to the carbon stability of the soil samples. Okay, so in conclusion, this study proves that the carbon stability of most of the archived soil samples from SQMP 2019 to 2020 have moderate carbon stability. Then after implementing the three-factor ANOVA, there is no statistically significant difference with 95% confidence interval, p-value greater than 0.05 in each treatment group based on the absorbance from the wavelengths with moderate carbon stability and even in the high and low carbon stability groups. In this case, there is not enough evidence or data to reject our null hypothesis. So this indicates that carbon stability will remain stable regardless of slope, depth, and tillage level. So that's all, and thank you for listening.